Hi everyone, can you hear me? Okay. Hi, so my name is Alexander Kim, and I work in the laboratory of Dr. Harris. And I'm just very pleased today to be able to present one of my projects that I've been working on, the identification of anti-NS1 antibodies for low-cost diagnostics. And in the Harris lab, what we study is dengue virus. Now, if you haven't heard of dengue virus, it is the most important arthropod-borne viral infection in the world. And it results, and it's the causative agent of dengue fever, which is also known as la quebradora, or breakbone fever. And that's so aptly named because the fever itself feels like your bones are breaking. And there are approximately 2.5 billion people at risk, which is greater than 40% of the world's population. There are 390 million infections that occur per year, and that number continues to increase as global trade, travel, and urbanization increases. Now, on this graph is a graph of a map from a Nature article published last year, and you can see the probability of occurrence of dengue in the world. So you can see primarily the subtropics and tropical regions are where dengue primarily resides, which is closely related to the Aedes mosquitoes vector. Now, what's important to realize also about not only that dengue resides within the tropics and subtropics, but also that most of these countries that reside in these areas are developing countries. So having a low-cost, rapid diagnostic available for these countries is extremely important in diagnosing dengue. Now, specifically what we're interested in is dengue NS1 protein. NS1 is just a non-structured protein. And why we're interested in it, there are out of 7 non-structured proteins that are encoded by the genome. It's the only one that is secreted. It is found as an intracellular dimer found at the cell surface membrane or as a secreted hexamer. It is implicated to be essential in viral replication, immune evasion, and vascular leak. And in our lab, what we have previously found was that NS1 vaccination of mice actually protects mice from lethal challenge of dengue, suggesting that anti-NS1 immune responses are essential for protection. And you can see on the bottom right, NS1, the crystal structure of NS1 that has just been solved this year. And it's really a great privilege to see it because after decades of research, we finally be able to see this. And so, in dengue infections, this is a graph of what happens in typical dengue infection. So, in the very beginning, you see a high level of diarrhea, as well as the presence of NS1, as early as day 1 of symptom onset. And then you can see the antibody response developing over time in IgG and IgM. Now, this is important to understand because current diagnostics have several issues with this. For example, viral culture is most accurate, but it's very time-consuming, 7 to 14 days. So by then, someone would already probably be past the acute illness stage where dengue is primarily present. And so PCR is sensitive and accurate, but it's also very expensive to run and presents an issue for these developing countries. Now, as far as immunological tests, they're much cheaper, but most are currently based on the detection of IgG and IgM antibodies, which you can see here are occurred later on during the dengue infection. So what we were looking at was looking at detection of NS1 as an indicator of dengue. So my project, or a lot of our projects, have been to develop a rapid, low-cost, accurate diagnostic tool for all serotypes and genotypes of dengue using a non-proprietary NS1 capture ELISA. Now, dengue has different serotypes, 4. You can think of them as different kinds of flavors of dengue. And within each kind of serotype, there are genotypes that further classify dengue. And it's important to realize that for our diagnostic tool, we have to be able to recognize all serotypes and all genotypes in order to be a reliable indicator of dengue. And if you don't know what an ELISA is, here's a quick demonstration of what it is. So specifically a capture ELISA, you have a plate about this big, each with 96 wells. And in each well, we coat first with a capture antibody. In the red is an antibody, and you can see the 2 prongs up there actually bind to proteins or antigens. So when we introduce NS1 into these wells, they actually bind to the antigen NS1. And then if we introduce a detection antibody, we can then bind to the NS1 that is bound to the capture antibody. And this detection antibody is particularly special in that it's conjugated to biotin. And biotin can bind to serpapin, which is conjugated to AP, which is alkaline phosphatase. The enzyme that converts PNPP into PNP, a yellow substrate. And by detecting this yellow substrate, we are able to, 1, qualitatively determine whether or not there is NS1, 2, and see whether or not the compare the relative amounts of NS1 in each flow. So in developing this diagnostic, we had several steps. So we first had to culture anti-NS1 hybridomas, which are immortalized antibody-secreting cells, and then purify monoclonal antibodies from them. And once we have a whole array of monoclonal antibodies, we can then maximize sensitivity by optimizing antibody pairs, so for the capture and for the detection. So by seeing which antibody pairs would produce the best result for our ELISA, we can then standardize ELISAs and then demonstrate highly sensitive cross-reactivity to NS1 from all the dengue serotypes and genotypes. And once we have that, we can then test it in the field for clinical samples obtained during the Nicaraguan dengue epidemics, and then evaluate the sensitivity and specificity of our assay. So here's a list of human antibodies that have been tested for serotype cross-reactivity. You can see that DB16 and DB115 were the only human antibodies that were able to cross-react from NS1 produced from all 4 serotypes, while for the mouse antibodies that have been tested, 5 of them were able to cross-react to dengue NS1 from all 4 serotypes: 711, 2B7, 9NS1, 3B12, and 391G4. Now, using these antibodies that were able to cross-react, we then started optimizing the antibody pairs, which one would produce the best results. So here's an example of an ELISA readout. So in this ELISA readout, on the y-axis, you have absorbance at 405 nanometers. And then on the x-axis, you see NS1 concentrations. These NS1 concentrations have been diluted twofold, as you can see, which decreases signal as you would expect from the ELISA data. The NS1 that we specifically used in this assay was obtained from a company called Native Antigen, who specializes in producing recombinant NS1. And so once we had these reagents in place, we found that the limit of detection was at 7.8 nanograms per ml. And we obtained that by multiplying the background by 2. And it's important to also note that during a dengue infection, the concentrations of NS1 can differ from anywhere from 10 to 100 nanograms per ml. And it usually depends upon, one, the serotype of the NS1, at what point in the infection it is in, and 3, the individual themselves. So, now that we were able to demonstrate that this indeed reacts with NS1, we then went about purifying NS1 from different genotypes within the different serotypes. So here's a list of viruses that we've used to infect a monocytic cell line called U937 DC sign cells. And because NS1 is produced and secreted, we can then collect NS1 from the supernagens of these cells and then attempt to purify out NS1 from these samples. So we're able to confirm that we're able to detect NS1 from our samples. As you can see in another ELISA readout, this is just some of the data we obtained, but all of them were able to react to the NS1. And so you can see it follows a dose dilution-dependent curve, and FT is just flow-through, which is with our negative control. But in order to completely evaluate our assay, we also, such as the limited detection we performed previously, we also need to make sure our samples are actually pure to make sure that the entire protein concentration is reflective of NS1. So we ran a silver stain, which stains for all proteins in our sample. And what we found was that there's also some proteins between 72 and 55, and you can see something at above 43 kilodaltons. And on the right, you can see a Western blot, which detects a specific protein in our sample, in our case, NS1. So you can see that around the 48 kilodalton marker, NS1 is present. But we also see some other proteins between 55 and 72 kilodaltons, which indicates that our protein samples need more purification steps to undergo before we completely perform a limited detection for these samples. Now, once we do perform this limited detection, we can then test clinical samples to make sure that our test is working for patients, and then continue to evaluate the sensitivity and specificity of our assay. I'd just like to acknowledge the Harris Lab, Dr. Eva Harris, Dr. Angela Green for guiding me through this entire process, Maria Jose, who has really taught me everything about NS1 and this project, Alexandros, the resident MD student, Dr. Henry Nelson, he's a great, awesome person. Mildred Galvez, the lab tech here, Louis, of course, the undergrad here, and everyone else that I just didn't mention. It's a Surbhi Fellowship, of course, and Rose Hills for providing funding for this project.
Any questions?
In the last slide you showed where you had the western blot and the silver stain next to each other.
Yeah.
Yeah, so it does look like there's a lot of other stuff on the silver stain that you're not detecting with the western blot, but do you think there could be any other proteins that form interactions with NS1 that you're just picking up when you verify it?
Right. So we initially went about thinking that maybe like there's also an envelope protein, and it'll be really strange. We don't know anything that really co-precipitates with NS1 per se, but we tried staining for E, which is typically around that 70 kilodalton marker, but that didn't turn out so well. It didn't stain. So we're currently trying to figure out what it exactly is.
NS1 has a lot of interacting partners inside the cell, but this was actually from what was secreted from the cell. So it's not a cellular protein that could be interacting. So mention that for yet.
Does the fact that it hexamerizes or polymerizes affect your readout? Or like, for example, like maybe for the protein sterile, like NS1 is just the monomer, but the hexamer is like 10 to 1 ratio, so that you have a lot of really pure hexamer actually somewhere.
Yeah, we looked at that, but the hexamer typically resides a little bit above, like around 130, 170, I think. So it wouldn't.
And would you be able to detect the hexamer with the western blot, or no?
I don't know if it's specific enough for that.
Well, it's more a question of whether if this is denaturing conditions,
Right.
then it would denature into the monomer.
Right.
Is there silver stain denaturing?
Yeah, so those
Same-
went on a
same
nature.
gel.
So you imagine doing ELISA as a diagnostic in the future, or what do you see?
Right. So we want to kind of aim towards having a rapid test, or kind of like having it as a diagnostic.
There's actually a lot of NS1 ELISAs out there and rapid tests, but their price point is so high that no developing country public sector can use them. So they're bought by clinical labs in developing countries, and like we can't even use them in our studies because it's too expensive. So we got some funding to kind of, in a way, reinvent the wheel and just make it available for the public sector in Latin America and beyond. And we're in IP discussion. I mean, then we have some people doing like a landscape analysis to see whether they want to move it into the rapid test or stay as an ELISA.
Isolate devices, or
Yeah, and with PATH, for instance, and we're looking at a number of different options. But the main thing is to just get something cheap in the hands of the public sector, because otherwise it's like all this fancy stuff is there and no one can use it.
I'm not sure I missed this, but is NS1 a protein that's specific to the dengue virus, or is it something that's...
So NS1 is produced by, there's multiple viruses that can produce like NS1, like yellow fever virus, West Nile virus, but for us, this is, NS1 is, we're like a 70% homology between serotypes, so we're using that as
Now, just one thing to mention, this is a really nice project because it's been done completely, I've been working for over 25 years in Nicaragua, and that's part of our vision, is to really work collaboratively. And so this was actually started by, and it's in complete collaboration with Maria Jose Vargas, who was mentioned, and she actually came up from Nicaragua and kind of ran the project with Alexander. Now she's back in Nicaragua doing the clinical assays to see if actually doing this with ELISAs on clinical samples. It actually looks much better. I don't know if I've told you this, but much better than the commercial kits right now.
Okay.
So I mean, it still has quick wares to go, but it's like it's comparing really well at the moment. So there you go. Thank you so much, Alex. That was awesome. So we're going to move on, and Magna Esarapa is going to speak next. She sets up, I'll introduce her. So she is also in molecular and cell biology, and she also has a minor in this in South Asian studies. And I should mention before, actually, Alex is working with Benji Green here, who's been her postdoc mentor, and he's been in the lab since May of 2012. And right after him, Meghna joined the lab in September of 2012, and she's, where's Purnima? And Parameswaran, who's her postdoc mentor. And she'll be talking about cell-free nucleic acids as biomarkers for shock in human dengue.
Hello. Can you hear me?
Ready to rock and roll?
Okay. Hi, so my name is Meghna Esarapa, and like Dr. Harrison, I'm an undergraduate in her lab. And my summer research project focused on the use of cell-free nucleic acids as biomarkers for shock in human dengue. And so like Alex mentioned before, dengue virus is a mosquito-borne flavivirus that places about 3 billion people at risk annually in tropical and subtropical potentially lethal forms of dengue hemorrhagic fever and dengue shock syndrome, represented by the tip of that pyramid, and one that unfortunately that poor child has in the picture above. And so currently there are no vaccines or therapeutics available to combat dengue virus, and other than worsening clinical symptoms, such as persistent vomiting or abdominal pain, there really is no easy way to determine whether an individual will develop severe dengue. And so the discovery of a biomarker as a prognostic tool would be very useful, and would definitely improve healthcare of dengue patients in endemic countries, which, as Alex mentioned earlier, tend to be developing countries. So one class of biomarkers that has proven useful in other fields are cell-free nucleic acids. And so nucleic acids are DNA and RNA, which you usually find within your body cells, but sometimes, as you can see in this figure, due to cell death or secretion, these nucleic acids are released into the bloodstream. And so some studies have shown the potential uses of these cell-free nucleic acids, such as to detect cancer progression or to screen for cancer. And this is because the cell-free nucleic acids in the blood of cancer patients tend to have mutations or other alterations that are quite characteristic of cancer. And another example is in pregnant women, the cell-free nucleic acids of the fetus will make its way into the mother's bloodstream, and that can be used to diagnose fetal aneuploidy, which can result in genetic disorders such as Down syndrome. And so my research hopes to investigate whether specific cell-free nucleic acid signatures in patient serum can serve as biomarkers to distinguish between mild and severe dengue disease. And specifically, I want to see if there is a difference between dengue severities. Specifically want to see if we can detect differences in the rates between dengue severities that map to dengue virus itself, potential other known pathogens, unknown pathogens, and human signatures. And there have been previous studies for biomarkers in dengue severity, but these focused on gene expression in blood cells or protein markers in serum. And one study actually looked at, found elevated levels of cell-free DNA in dengue patients, especially severe dengue. But like I said, my research hopes to focus on specific signatures that differ and correlate with dengue disease severity rather than just the levels of nucleic acids. So the samples we're using are from Dr. Harris's hospital study in Nicaragua, and these serum samples were taken on the first day of hospitalization of patients with either dengue fever, which is represented by the mild dengue, or dengue shock syndrome, represented by the severe dengue. And so these samples also represent all serotypes of dengue virus, because the point is to find a biomarker that can work across all dengue infections. And so we have 19 mild dengue samples and 20 severe dengue samples, and we're using water as a negative control. And for statistical purposes, we have processed these samples in two different batches, the first being the training set and the second being the testing set. And so we developed the following protocol to acquire the cell-free nucleic acid signatures from patient serum. And so our first step was to extract the total nucleic acid content from the serum, and this includes both DNA and RNA, and we use a Kaya-AMP nucleic acid extraction kit to do so. And then the next step involved reverse transcription, which created complementary DNA from any RNA in our samples. And so we did this without primer and with the random hexamer primers, because the point of the study is to find the sequences that exist in the serum, so we do not have one specific sequence we want to amplify, and the use of these random primers ensures coverage of all potential sequences within our serum samples. So the next step was multiple displacement amplification, and so this involves an enzyme called Phi29, which is able to amplify very low levels of DNA of various lengths with very little error, and this makes use of the branching method, where the Phi29 creates a complementary strand, as you can see in this figure, which then itself immediately becomes a template for further priming and synthesis events. And so at the end of this, we have pools of double-stranded DNA. And so then we use an Illumina Nextera kit to prepare libraries for sequencing. And so we set off our libraries to the QB3 facility here on campus, and we used more learning machinery to perform high-throughput sequencing on our samples. And so this basically resulted in millions of reads at very low cost. And in fact, the price per read turned out to be 0.003 of a penny, which is pretty cool. So once we had our sequences, some bioinformatics was necessary in order to make sure we were analyzing true reads rather than just artifacts of our sequencing process. And so for our first step was to filter for high-quality reads. And so we used the quality scores for each nucleotide that we got from the sequencing. And we also made sequences with greater than 50% variety of organisms, and we wanted to make sure that there was no confusion in determining where a sequence arose from. And then we mapped our reads using an aligner called Bowtie 2. And so we aligned them to the nucleotide reference database in the National Center for Biotechnology Information website. And this contains the genomes of all sequence organisms. And we also aligned them to the genomes of the 4 different dengue viral serotypes. And so once again, we filtered for high quality mapped reads. So this meant that we retained the reads with the highest mapping score, which meant that these reads that we retained, at least 50% of their read length mapped to some type of reference sequence. And then we also further collapsed the map reads to retain one unique identity of over-amplified reads. And lastly, for each read that we had, we retrieved taxonomy information and common gene names, and then we further proceeded to summarize these hits according to various categories, as you'll soon see. And so the data analysis is still ongoing, but the following is an example of what we've been doing. And currently I'm analyzing the first training set samples, which consists of 10 mild samples, 10 severe samples, and 1 water control. And so first I wanted to see what percent of reads in each sample actually mapped to a known reference sequence. And so as you can see here, the water actually had a lower percent of map reads, which is to be expected because, and it's good to see because water doesn't have much nucleic content in it in the first place. And we also saw a lower percent of map reads for the shock cases. And this could possibly be because the shock cases contain novel sequences that are not yet cataloged in the reference database, or it could also be because, like the water, it had less nucleic acid content in the first place to begin with. And so next we wanted to see what percent of reads in each sample mapped to one of the superkingdoms. So if you look at this graph, each circle marker represents one sample, and in each plot you should see 10 different colors to represent the 10 different samples. And so the y-axis is the percent relative abundance of reads in each sample that maps to one of the superkingdoms on the x-axis. And so if you look closely, you'll see that the majority of reads in the highest percent of relative abundance of reads for the majority of reads in the mild cases and shock cases, they mapped to the eukaryotic superkingdom versus the water control, where the highest percent relative abundance of reads is found in the bacterial superkingdom. And once again, this was really good to see because the nucleic acid content of human samples, human serum, versus water should be different. And so then we further wanted to see what percent of reads in each sample mapped to a specific virus. And so the x-axis contains all the different viral species that we found among our different samples. And once again, the y-axis represents the percent relative abundance of reads in each sample, represented by one of the different markers that map to one of the species on the x-axis. And so there was a very clear difference in the viral profile between our human samples and the water control. And this is especially noticeable in dengue virus, which is completely acid in water, but actually pretty prominent in the mild and shock. And so then we took a closer look at the difference in percent relative abundance of dengue virus in severe versus mild cases. We saw a trend where only 3 out of the 10 mild cases had dengue reads versus 7 out of the 10 shock cases which had dengue reads. And this was a substantial difference, and it was nearly statistically significant. We still have to analyze our second set of samples, so there might potentially be a statistical difference once we combine the data. And lastly, we also wanted to see where the dengue reads in each sample map to the dengue virus genome. And so each sample is separated by a red line, and the top chunk represents the 3 mild cases of dengue reads, whereas the bottom chunk represents the 7 severe samples with dengue reads. And so if you consider the entire stretch of that screen to be the dengue virus genome, each dash or line means that that particular area of the genome is represented by a read in the sample. As you can see, there is actually pretty good coverage of dengue virus, of the reads of the entire dengue viral genome, and especially in the shock cases. And so this let us know that the dengue reads we were looking at were not just over-amplification of one read, but rather represented the entire genome. And it was also supported our earlier idea that dengue would be more highly represented in the shock cases. And so, summary of this, when we mapped the reads to different superkingdoms and viruses, we saw that the profiles differed between the human samples and water control. We also noticed a substantial difference in the percent relative abundance of dengue in mild and severe saw that the dengue rates showed pretty good coverage of the genomes of the different dengue serotypes. And so for the future, we will continue analysis, and more than just dengue, we also want to see if there are any differences in reads in known pathogens or unknown pathogens, and also human signatures. And we'll also later analyze the second set of samples and see if we can match the patterns with the first. And for all your help, I thank Dr. Eva Harris, my amazing mentor, Dr. Karan Parashuran, Surbhi Travedi, the Harris Lab, and of course their LMS for providing me this amazing opportunity.
Any questions? I'll start off with one. When you say the water control, really what that is reflecting is all of your reagents, right? I mean, so because I remember talking about some of the bacteria or others can be coming from the enzyme press, because it's essentially going through the whole system. It's not just water.
It's not just water.
Going through the process.
We started from the top. We actually extracted nucleic acids from the water itself and bring it through the entire method.
Yeah. So it's probably like where are all those sequences coming from? It's probably from
That reagent.
yeah, and that's why the reagent was